Hello friends. In this lecture we'll talk about enzymes. The enzymes topic has been divided into two lectures. So we'll discuss some of the aspects of enzymes in this lecture and the remaining in the next lecture. Now here are some questions which have been asked in the previous CSR examinations. Number one, enzymes accelerate a reaction by which one of the following strategies? So here four options are given. Number one, decreasing energy required to form the transition state, increasing kinetic energy of the substrate, increasing the free energy difference between substrate and the product, and fourth option is increasing the turnover number of enzymes. Another question which was asked in June 2017 was based on the action of a serine protease such as trypsin, chymotrypsin, which are one kind of serine proteases. So here the peptide sequence is given, which is acting as the substrate, and the serine protease site of action is shown where the protease cleaves the peptide. And in this column, the kcat is given and km values are given, and three statements are given. And out of these three statements, which statement is correct, that you have to identify, and from the given options which correct combination is there. Okay, so you have been asked that which of the following combinations of the above statements is correct. So you have to have information about the kcat, then km, what is its significance, then from this data what else you can identify or what else you can analyze, that you have to know. So let us first discuss about some of the basic concepts required for this understanding. So here enzymes are the biocatalysts which speed up the reaction rate without getting transformed permanently. Almost all the biochemical reactions that occur in the cells are catalyzed by enzymes. These enzymes are generally proteinic in nature. The exception is ribozymes, which are the catalytic RNA molecules. Some of the antibodies also have catalytic activities, and these are called as enzymes. The reactants on which the enzymes act, they are called as substrate, and the substrate are then transformed into product. Enzymes are highly specific in their action, and these exhibit enormous catalytic power. Enzymes increase the rate of reaction by lowering the activation energy which is needed by the reactants to attain. So here the reactants are substrate. They change only the rate at which equilibrium is achieved. They have no effect on the position of the equilibrium. So these enzymes, they can be either simple enzymes or conjugated enzymes. Enzymes which are simple in their nature, they are generally made up of only proteins. Whereas conjugated enzymes, they contain a non-protein group called as cofactor, which is required for the biological activity. Here, the inactive form of enzyme is called as apoenzyme, which in presence of cofactors becomes a complete enzyme called as holoenzyme, which in turn is active in nature. So here the cofactors can be of different types. They can be simple metal ions such as copper ions, zinc ions, molybdenum ions, and so on. They can be also organic in nature, like coenzymes. Coenzymes, they are the non-protein component which are loosely bound to the apoenzyme by non-covalent bond. The examples include several vitamins or compounds derived from vitamins. Prosthetic group is another kind of cofactor. This is also a non-protein component which is tightly bound to the apoenzyme covalently or non-covalently. These are called as prosthetic groups. Examples include heme, flavin, biotin, iron sulfides, copper, and ubiquinone. These are some examples of the prosthetic groups. So here the list of several cofactors required by the different enzymes are listed. Copper ions, they are required by the cytochrome oxidase. Ferrous or ferric ion, they are required by cytochrome oxidase, catalyst, peroxidase, and so on. Potassium ions are required by pyruvate kinase. Magnesium ions are required by hexokinase, glucose 6-phosphatase, pyruvate kinase, and so on. Here, the different coenzymes are shown. For example, the biocytin is the coenzyme which is involved in the transfer of the chemical groups, carbon dioxide, okay, and the dietary precursor in mammals is biotin. So from biotin, this coenzyme can be made available. Coenzyme A is another coenzyme which is involved in the, or which is required by the enzymes that are involved in the transfer of acyl groups, and these can be obtained from pantothenic acid and other compounds. Likewise, flavin adenine dinucleotide, it is involved in the transfer of electrons, and the dietary source is riboflavin or vitamin B2. So you can have a look at several coenzymes which are involved in the transfer, or which are required by the enzymes, which in turn are involved in the transfer of several chemical groups, and from which sources they can be obtained, that is listed in this table. If we talk about the nomenclature and classification of enzymes, the naming of the enzyme in majority of the cases they end in ase, where for example sucrase catalyzes the hydrolysis of sucrose. The name may also describe the function of the enzyme. For example, the oxidases, they catalyze the oxidation reactions. Sometimes common names are used particularly for the digestion enzymes such as pepsin and trypsin. Some names describe both the substrate and the function. For example, alcohol dehydrogenase, which oxidizes ethanol. Classification of enzymes is done into 6 functional classes, which is referred as EC classification or Enzyme Commission number classification by the International Union of Biochemists IUB on the basis of the types of reactions which they catalyze. So these enzymes can be broadly classified into 6 major classes: class 1, 2, 3, 4, 5, and 6. So class number 1 includes all the oxidoreductases, class 2 includes all the transferases, class 3 includes all the hydrolases, class 4 includes all the lyases, class 5 includes all the isomerases, and class 6 includes all the ligases. So here, these class numbers you must remember in the sequence. So there is one illogical formula to remember the sequence of the classes. So here you just remember this term Utlaili. Okay, so Utlaili will help you to remember the sequence of the classes of these enzymes. So let us have a look what are these enzymes doing in each class, or which class of which types of enzymes are included in different classes. Oxidoreductases, these include all the enzymes that are involved in oxidation reduction reactions. Transferases, these are the enzymes which are involved in the catalysis of the transfer of functional groups. Hydrolases, these include all the enzymes that bring about the hydrolysis of various compounds, and lyases are the enzymes which are specialized in the addition or removal of water, ammonia, carbon dioxide, etc. Isomerases include the enzymes that are involved in the isomerization reactions, whereas the ligases, these include all the enzymes that are involved in the synthetic reactions or bond forming reactions, where two molecules are joined together, and here the energy compound is used, that is ATP may be used in this case during bond formation. Each class in turn is subdivided into many subclasses which are further divided. A 4-digit enzyme commission number is assigned to each enzyme representing the class, which is the first digit, subclass, which is indicated by the second digit, subclass, which is indicated by the third digit, and the individual enzyme, which is indicated by the fourth digit. So, for example, alcohol dehydrogenase, its enzyme commission number is EC 1.1.1.1, whereas phosphofructokinase, its enzyme commission number is 2.7.1.11. So, this indicates that phosphofructokinase belongs to class number 2, subclass 7, subclass 1, and it is the 11th enzyme in its category. Enzymes, they lower the activation energy. So here, for the reactants to get converted into substrate, these reactants must obtain the energy which is called as activation energy, and after obtaining the activation energy they get excited, and after that the reactants are converted into product. So this is the curve which indicates how the reaction will proceed in absence of a catalyst or an enzyme. Whereas in presence of enzymes, the reactants, they require less energy to get activated. So activation energy is lowered in presence of enzymes, because of which the reaction occurs in a faster manner. Reaction proceeds in a faster manner, and you can see the delta G, or that is the free energy change of the reaction, is unaffected by the enzyme. So here, only in presence of enzyme, the activation energy that is needed by the reactant to attain, that is lowered. So only this much energy is required for the reactants to attain in presence of enzyme, and that's why the reaction proceeds in a faster manner in presence of enzymes. Active site of the enzyme. Enzymes are larger in size compared to substrates which are relatively smaller. Evidently, a small portion of the huge enzyme molecule is directly involved in the substrate binding and catalysis. The active site or active center of an enzyme represents as the small region at which the substrates bind and participates in the catalysis. So here you can see the active site is shown, where the substrate will bind, and these are the binding groups, whereas some of them are the reacting groups. So reacting groups must bind to the catalytic site, whereas these are the binding sites within the active site where the binding groups are occupied. So likewise, the active site of the enzyme will contain the binding site for the binding groups and catalytic sites for the binding of reacting groups of the substrate. When the enzyme and substrate are involved, so here the substrate binds to the enzyme and the enzyme substrate complex formation occurs. So this enzyme substrate complex is an intermediate, and during on this, the substrate is then transformed into product, and the product is then released. After release of the product, the free enzyme is again made available to bind to the another substrate. So enzyme substrate complex formation is necessary for the conversion of substrate into product by the enzyme. Mechanism of enzyme action. To explain the mechanism of enzyme action, three theories are proposed: lock and key hypothesis, induced fit hypothesis, and transition state stabilization theory. So here, lock and key model indicates how the enzyme binds to the substrate. The active site has a rigid shape according to this model, and only substrate with the matching shape can fit into it. The substrate acts as a key which fits into the lock of the active site. Now this model is a very older one and does not work for all enzymes. So here is the figure which is indicating that how a substrate can fit into the enzyme like a key into the lock, and the binding groups are occupied in the binding site, the reacting groups of the substrate are occupied in the catalytic site, and the conversion of substrate and product can occur if the reacting groups are occupied in the catalytic sites. The another model is proposed. In the induced fit model of the enzyme, the active site is flexible but not rigid. The shape of the enzyme, active site, and substrate, they adjust to maximize the fit, which improves catalysis. There is a greater range of substrate specificity, and this model is more consistent with a wider range of enzymes. So here the enzyme has a slightly different structure with respect to its active site, and upon binding of the substrate to the enzyme, some changes may occur in the structure of enzyme, because of which the favorable interactions between the substrate and the enzyme occurs, resulting in the binding or bringing the catalytic site near to the reacting groups, and the substrate now can then be converted into product by the enzyme. Now, the another theory was proposed. Now, this induced fit model is not able to explain much that how just input of less energy can result in conversion of substrate into product, and that too at a faster rate. So here, to explain that, the transition state stabilization theory or strain theory was proposed to explain that thing. So here, initially when the substrate binds to the enzyme substrate complex formation occurs, and during this initial interaction, there is unfavorable interaction between the substrate and the enzyme. But later on, these unfavorable interactions, they get diminished, and the favorable interactions occur between the substrate and enzyme, and the substrate is also subjected to certain bending or just stretching of the certain functional groups, which results in weakening of the bonds at certain locations on the substrate. That makes easier for the enzyme to convert substrate into product, and that is how the speedily the substrate can be then converted into product. Units of enzyme activity. Enzymes are quantified in terms of their catalytic activity. Enzyme activity is measured as the amount of substrate lost or product gained per unit time. So either you can monitor the disappearance of substrate or the appearance of product. The unit used to report the enzyme levels are activity units. So this activity can be expressed as international units or catal. International units. Enzyme unit or international unit is defined as the amount of enzyme activity that catalyzes the conversion of 1 micromole of substrate per minute under standard assay conditions. So here 1 international unit is 1 micromole per minute. Catal is also referred as the SI unit of enzyme activity. So in order to maintain uniformity in the expression of enzyme activities as units worldwide, the Enzyme Commission of IUB has suggested SI unit to be used. A new unit, namely Catal, which is abbreviated as CAT, was introduced. According to this, 1 cat denotes the amount of enzyme required for the conversion of 1 mole of substrate per second under standard assay conditions. So this activity of the enzyme may also be expressed as millicatals, microcatals, nanocatals, and so on. Specific activity. To minimize the errors occurring due to the variation in the concentration of enzymes in enzyme sample, units of enzyme activity may be related to the total protein content of the sample being assayed, rather than giving weightage to its weight or volume of the sample used in the enzyme assay. Amount of enzyme per milligram of protein required to convert 1 micromole of substrate to product per minute under standard assay conditions is called specific activity of enzyme. It is expressed as international unit per milligram of protein or catals per kg of protein. Let us have a look at the kinetics of single substrate enzyme catalyzed reactions. Leonard Michaelis and Maud Menton, while studying the hydrolysis of sucrose catalyzed by the enzyme invertase, proposed hypothesis or Michaelis-Menton's hypothesis with certain assumptions. So here, Michaelis-Menton's assumptions for single substrate enzyme catalyzed reactions are noted. Number 1, only a single substrate and a single product are involved in such reactions. So here this assumption involves that the single substrate and single enzyme is involved in the enzyme catalyzed reaction. The process proceeds essentially to completion. The concentration of the substrate is much greater than that of the enzyme in the system. An intermediate enzyme substrate complex is formed, and the rate of decomposition of the substrate is proportional to the concentration of enzyme substrate complex. Michaelis-Menton's equation was modified by Briggs and Haldane, which is referred as Briggs-Haldane modification. So here the enzyme substrate reaction is shown, which reacts to with the enzyme reacts with the substrate to form enzyme substrate complex, and the enzyme substrate complex is from that the substrate is converted into product and the product is released. So here the rate constants are mentioned, and the rate constant which dictates the enzyme activity or enzyme rate is the K2. K2 is the rate limiting constant for the conversion of substrate into product. So here, according to Michaelis-Menten's equation, the initial velocity of the enzyme V0 is equal to Vmax into S0 upon S0 plus Km, where V0 is the initial velocity, Vmax is the maximal velocity, S0 is the initial substrate concentration, and Km is the Michaelis-Menten's constant. This formula is there at constant E0, that is the initial concentration of enzyme. Now here, according to Briggs-Haldane steady-state assumption, the enzyme substrate complex which is formed almost remains same at the steady state. So here a steady state is achieved between the enzyme and substrate. When the substrate is present at very high concentration, all the enzymes will get saturated with the substrate, and the enzyme majorly will be present in form of enzyme substrate complex. So you can see over here during the initial stage of the reaction, there is less product formation, but as the steady state is assumed, now the substrate is getting converted into product. The number of free enzyme later on then also remains constant, which will be very negligible once the enzymes are saturated with the substrate, because substrate concentration is very high compared to the enzyme concentration. If we plot the substrate concentration versus initial velocity of the enzyme, we get a hyperbolic curve. Now, according to this, as the substrate concentration increases, the reaction rate also increases and it reaches to a maximal value, which is referred as Vmax. Now, if we look at the half of the Vmax value and plot it onto the curve, we drop it onto the x-axis, we get the Km value. So this intercept on the x-axis is referred as Km, that is the Michaelis-Menten's constant. Significance of MM equation: number one, the Km value, which is the Michaelis-Menten's constant, it is defined as the substrate concentration which gives an initial velocity equal to half Vmax value, and Km will have the same units as initial substrate concentration, S0. Km is independent of enzyme concentration and is the characteristic of the system being investigated. Km indicates affinity of enzyme towards substrate. High Km means low affinity, whereas low Km indicates high affinity of enzyme towards substrate. It can thus be used to identify a particular enzyme. In most cases, Km values lies in the range of 10⁻² to 10⁻⁶ moles per liter. Turnover number is another characteristic of the enzyme. Here it is designated as Kcat. It is obtained from the expression Vmax is equal to Kcat into E0. For single substrate enzyme catalyzed reactions, Kcat is the K2, that is the rate constant, which is involved in the conversion of enzyme substrate complex into E plus B. For complex reactions, Kcat will be the function of individual rate constants. Catalytic efficiency, which is designated as Kcat upon Km, it is also referred as specificity constant. A high value approaching that of K1 indicates that the limiting factor for the overall reaction is the frequency of collisions between enzyme and substrate molecules. A low value would be more in keeping with the equilibrium assumption. According to this, the greater the ratio, higher the rate of catalysis is. Conversely, the ratio, if it is lower, the catalysis is slower. A comparison of Kcat upon Km for alternative substrates can also be used as a measure of the specificity of an enzyme. A diffusion limited enzyme catalyzes a reaction so efficiently that the rate limiting step is the substrate diffusion into the active site or product diffusion out. Kinetically perfect enzymes have a specificity constant Kcat upon Km on the order of 10⁸ to 10⁹ per mole per second. The rate of the enzyme catalyzed reaction is limited by diffusion, and so the enzyme processes the substrate well before it encounters another molecule. Now, to get the accurate Km value, we use the LB plot, which makes use of the MM equation. The MM equation is inverted on both the sides, reciprocal is taken, and the LB equation is shown over here. So 1 by V0 is equal to Km upon Vmax into 1 by S0 plus 1 by Vmax. According to this, the intercept on the x-axis indicates the Km value, whereas the intercept on y-axis will give idea about the Vmax value, and the slope is Km upon Vmax. So this equation is like that of a line, and so the LB plot is a linear one. So from LB plot, we can accurately estimate the Km and Vmax values. Factors affecting the enzyme activity. So here several factors affect the enzyme activity, concentration of enzyme, substrate, temperature, then pH, product concentration, and so on. So here, as the enzyme concentration increases, the enzyme velocity also will increase in a linear manner, whereas in case of substrate concentration, we have already seen a hyperbolic curve is seen. As the substrate increases, initially the reaction rate also increases, and then it gets constant because the enzyme concentration is kept constant. Temperature, as the temperature increases, the reaction rate also may increase, and we get a bell-shaped curve. So at a particular temperature we'll get a optimum activity, whereas further increase in the temperature can result in decrease in the enzyme activity. So here that may be due to the inactivation or degradation of the enzyme due to high temperature. Q10 value is very important in this case, which is also referred as temperature coefficient. It is defined as the increase in the enzyme velocity when the temperature is increased by 10 degrees Celsius for a majority of enzymes. Q10 is 2 between 0 degree and 40 degree. Increase in temperature results in higher activation energy of the molecules and more molecular collision and interaction for the reaction to proceed faster. Effect of pH is another factor which affects the activity of enzyme. So according to this also, as the pH increases, the reaction rate also may increase and tends to be a maximum or optimum at a particular pH, and the later on the increase in the pH may result in drop in the activity. So that changes in the pH may affect the structure of the enzyme. The enzyme's functional groups may get protonated or deprotonated, because of which the repulsion of between the like charges can result in the disturbance in the conformation of the enzyme. So let us have a look at certain questions which were asked. Enzymes accelerate a reaction by which one of the following strategies? Decreasing energy required to form the transition state, increasing kinetic energy of the substrate, increasing the free energy difference between the substrate and the product, or increasing the turnover number of enzymes. So here we see that the decreasing the energy required to form the transition state is the correct one, because they lower the activation energy. Next one, here the peptides are given and the kcat and km value are shown. Now from this, what you have to do, just count the number of amino acids that are present and calculate the kcat upon km value. So here you can see these peptides are of having 3 amino acids, these are having 4 amino acid long peptide, this is having 5 amino acid long, and so on. Kcat upon km value, when you calculate, you will get this, and from this you can get the idea that here the kcat upon km value is increasing as the peptide size is also increasing. Okay, so but here now you can see that the valine is there, and because of that the kcat upon km value is slightly different. So here what are the options given? Catalytic efficiency increases with the size of the peptide, then yes, it is right. Amino acid at the hydrolytic cleavage position of the peptide is critical. Yes, it is right. And catalytic efficiency decreases from 3 amino acid peptide to 4 amino acid peptide. That is wrong. Okay, so it is increasing actually. So the correct option is this A and B. So with this, we complete the discussion on enzymes. Thank you.